time. Please be seated. You can.
Good afternoon, all of you. Also welcome to the public audience. And I open hereby officially this academic ceremony in which Ms. Fang Wang will defend her academic thesis entitled Targeting CD36 and v-ATPase for Diabetic Cardiomyopathy Treatment. May I invite you to present a summary of your studies and the conclusion of your thesis in the next 15 minutes, and I'd like to give you the word.
Thank you, dear director. Now I can start, right?
You can start, yes.
Okay. Dear prorector, members of Corona, friends, families, and supervisor team, in the next 15 minutes, I will give the summary and conclusion of my thesis entitled Targeting CD36 and v-ATPase for Diabetic Cardiomyopathy Treatments. To understand this, I would like to start with the heart. As you can see, the heart's continuously beating from the beginning of life to the end. The contractility of this heart brings energy to the whole body. To maintain its contractility, it needs energy. The energy comes from mainly fats, but also sugar and others like ketone bodies, amino acids, and lactate. The energy source normally comes from our diet. When you eat meat or rice, you get you get sugar, and when you eat fat-enriched foods, you get fats. However, but in order to keep a healthy diet, the vegetables and fruits are also included in the normal diet. However, due to the changes of lifestyle, mainly Western diets, people are more exposed to high sugar enriched diets or high fats enriched diets. This causes problems of obesity. Obesity puts people into a risk of developing diabetes, and people with diabetes are more likely to develop heart disease at a younger age, and also higher chance to die of heart disease. The term that we focused is diabetic cardiomyopathy. It's the very major complication of diabetes, and it's the phenotype of this disease is described as you can see the left bottom of this heart is very much enlarged, and the muscle of this part is vacant. So the heart muscle is not strong enough to pump blood out of the heart to provide energy for the whole body. Thus the contractility is reduced, and it leads to a very unhappy heart. Thus heart failure occurs. And according to the report of WHO, and heart disease is also the leading cause of death globally. And as a scientist, the aim of my thesis is to find novel treatment strategies for diabetic cardiomyopathy. And next, I will give you a schematic presentation from the molecular level, what is the mechanism under the development of diabetic cardiomyopathy. So here is the blood vessel, and this is a heart cell, and the pink area is the cytosol, and the gray area is the membrane of this heart cell. Inside the heart cell, there's an intracellular organelle called endosome. So after a meal, the fats and sugar will be transported via blood flow to the heart cell, and it also secretes insulin to proceed the uptake of fats and sugar into the heart cell. The insulin needs to bind with the receptor, and the uptake of glucose, of sugar and fats will be done via the transporter, which is present on this membrane of this heart cell, but also inside this organelle endosome. The trafficking of this transporter between the membrane and this endosome promotes the uptake of this fat and sugar into the heart cell. So when insulin binds to insulin receptor, the transporter will go to the membrane, and next the fat and sugar will be taken up by the heart cell. And this will be further utilized for energy production for the heart cell. However, in the long-term overexposure of fats, there will be more transporter present on the plasma, on the membrane. There will be increased fat uptake in the heart cell, and this will lead to this will be sensed by v-ATPase. This is very important protein which is responsible for this endosome acidification. And this acidification is very important to keep this fat transporter internally. So more increased fat uptake in the heart cell will stimulate the disassemble of v-ATPase, of this v-ATPase. So the V1 subunit run away from V0, and it leads to increased endosome pH. And this causes more fat transport on the membrane. And it's increased again for the fat uptake in the heart cell. And it leads to, so here is a vicious cycle, and it leads to lipids, it leads to fat accumulation in the heart cell. This fat accumulation has some consequence. It's influence the insulin signaling, and it also leads to decreased contractility of the heart cell and leads to a unhappy heart. So we hypothesize that if we can target the translocation of this fat transporter, but directly, or we can keep the fat transporter internally by targeting the v-ATPase to rescue the phenotype of heart failure. Our first strategy is what nature brings us. We focused on the traditional Chinese medicine. It comes also from herb-derived compounds. It has been used in China for thousands of years in clinic. It has been proved beneficial in different contexts, importantly, it's also very abundant in our diet, such as berries. More importantly, it has been commercialized as supplements in the market, so people can take it freely. So we want to test if the TCM compounds is beneficial in the context of lipid-induced cardiomyopathy. So our experimental setup in this context is we took the cell lines from mouse. We also took the rats. We isolated the rat primary cardiomyocytes from the rats, and we give them high-fat diets. In the meantime, we also give them high-fat diet combined with these compounds. And our results showed that the TCM compounds leads to less fat presence on the membrane, less fat accumulation in the heart cell, better insulin response, and increased contractility of the heart cell. Thus, we can conclude that TCM compounds restores insulin sensitivity and contractility in fat-overloaded heart cell. Our next strategy is NMN. NMN is also another kind of supplement. It has been tested in different contexts and shown to be beneficial in different organs. However, the mechanism of NMN in the context of lipid-induced cardiomyopathy is not known yet. So we took the mice and we give them high-fat diets, also high-fat diets combined with NMN. And our results showed that NMN also leads to less fat transporter present on the membrane, less fat accumulates in the heart cell, better insulin response, and increased contractility of the heart. more importantly, we have found the mechanism of how NMN works. It promotes the reassemble of v-ATPase, and this leads to the endosome acidification. That's the reason that it keeps the fat transporter internally. Thus, we can conclude that NMN preserves the heart function during fat overload by preventing v-ATPase disassembly. Thus, in the lipid overloaded situation, the TCM compounds inhibit the translocation of this fat transporter, and the NMN stimulates the reassemble of v-ATPase, and this keeps the endosome more acidified, and those two strategies leads to more fat transporter internally, and leads to less fat uptake in the heart cell, less less fat accumulates in the heart cell, and better insulin response. More importantly, increase the contractility of the heart. That leads to a happy heart. In the end, I would like to summarize that from the heart failure to the happy heart, it's a long journey, and from my thesis, we can conclude that we found CD36 and v-ATPase are a suitable target for fat-induced cardiomyopathy, and TCM compounds and NMN supplementation are attractive nutrient strategies to combat fat-induced cardiomyopathy. Thank you for your attention, and now I would like to give the word to the prorector.
Thank you very much, and thank you for your nice and well-illustrated presentation. Good job, compliments with that. And before we start the discussion, I would like to present also to the audience, but also to our Corona team here, the promotion team. That's the normal procedure. And you do have a rather large promotion team with 4 members, which is substantial. And I would like to mention that we have here, and I just have to give a little order here. So we start from my left side, maybe it's from your right side, to Dr. Nabben. She is from the Department of Genetics and Cell Biology, followed by Dr. Neumann. He's from pathology, I'm sorry. And then we go to Dr. Luyken. He's also from the Department of Genetics and Cell Biology. And finally, the first promoter sitting here right in opposite of me, it is Professor Glatz, and he is involved in the aspect of metabolic aspects of cardiovascular diseases at our university. My name is Professor Steinbusch for the audience. I am the pro-rector today, and I'm leading this discussion. And now, without any further ado, we're going to start, and I received a list of opposition, and I would like to give the opportunity to the chair of your assessment committee, Professor Hesselink, who is he is chair of the Department of Physical Activity and Metabolic Health at the Institute Nutrim at our university, and he is able and he is willing to address you as first opponent. I give you the word.
Thank you, Pro-Rector. Dear candidate, it is my pleasure to congratulate you with the thesis that you're currently defending. I think it's a comprehensive piece of work that holds lots of experiments, lots of pivoting probably, and I'm quite sure it's taken hours and hours and hours in the lab and in the writing period. Congratulations with that, and I'd also like to involve your promotion team in these congratulations, because I think you've done a great job. But next to that, obviously, it's also the moment to ask questions on your thesis and have a little scientific debate. And I'd like to debate with you a bit on the role of lipids in what you call lipid overload-induced cardiomyopathy. From that title, I sort of deduce already that lipids, you consider lipids to be the bad guys in cardiomyopathy. Is that correct?
Highly esteemed opponents, thank you for your kind words and your question. Indeed, in my thesis, we consider that lipids are the bad guys in the lipid-induced cardiomyopathy.
Good. Well, there's a couple of questions that I have that you can basically answer, and I'd like to trick you to do that with its physiology or its pathophysiology. So the answers I would like to get to the questions are short, and you can either choose this is physiology or this is pathophysiology. And with that, I'd like to have a little bit of a debate. Okay? So in your review in chapter 3, you mention the consequences of lipid-induced cardiomyopathy, and I'll name a few. You say there's dysregulated PPAR signaling, there's mitochondrial dysfunction, insulin resistance might occur, there's inflammation going on, oxidative stress, and dysfunctional calcium handling. And I think you consider all these physiological or pathophysiological?
In the context of lipid overload, we consider all these consequences the pathophysiological of lipid overload.
That's what I was anticipating. And then we move to chapter 4, where you sort of play around with all these Chinese herbs, or maybe drugs, if you might call them like this. And you do that elegantly in a Petri dish. And I'd like to go to figure, I think it is figure 1 or 2 on page 61. Figure 1. Are you with me?
Yes.
Yeah. So in panel A, we see already stains of cardiomyocytes exposed to different compounds, and I think you start by overloading these with lipids in the HP condition, which I think is high palmitate. And then from panel B, we read a couple of bar graphs, and we see that upon the high palmitate, there is excessive lipid accumulation. Physiology or pathophysiology?
Pathophysiology.
Good. We move to panel C, where we see the effects of resveratrol on the amount of cell surface bound CD36. You choose 3 different panels to show, or 3 different bars to show. Low palmitate, and there we see that there is an induction of CD36 on to the membrane, a relative induction. We see high palmitate, and we see high palmitate and resveratrol. So with high palmitate and resveratrol, the effect that you observe, would you call that a physiological benefit, or is it a pathophysiology thing that's happening there? So I think it's maybe easier if you go to the bar graph at the right, where you basically see the insulin-mediated effects.
Yeah. It's the beneficial effects of the resveratrol, so it rescues the pathophysiological consequence of high palmitate.
Great. Same thing, I think, for quercetin in panel D. For, what is it, curcumin or which one? But either way, for E, F, and G, we follow the same thing with the different compounds.
Yes.
Yeah. Then in panel H, we see carbon-14 labeled palmitate uptake that is blunted in the high palmitate condition, and that seems to be rescued with resveratrol, with quercetin, with berberin, etc. Physiology or pathophysiology?
It rescues all these GCM compounds rescues the physiological consequence of high palmitate.
We move a little further and we go to figure 2, page 64. Similar setup, but now you measure insulin-stimulated glucose uptake. We basically see something similar as we observed with the palmitate uptake, right?
Yes.
So your compounds seem to rescue the glucose uptake that was hampered, the insulin effects that were hampered by the high palmitate condition. Physiology or pathophysiology?
I think it's the same as the palmitate uptake.
Good. So everything thus far we consider to be physiology.
Yeah.
Okay. Now I'd like to move to page 77, supplement 1. And there you need to help me a little. In 77, in figure 1, we do see the effects on cell viability.
Yes.
How did you measure cell viability?
Via the SRB assay.
Okay. So what I actually see is that in many of the high palmitate conditions, you use a range of concentrations of the different compounds that you have been using before. But if I look closely, or not really closely, but sort of through, yeah, the, what do you call it? Door mijn oogharen. If I look through my eyelashes, sorry for not getting the, then I tend to think that with these compounds you actually have reduced cell viability.
Yes, but only occurs in the high concentration of those compounds. And in the compounds, in the concentration that we used for all the assay that we did in these parts, they are not toxic for the cell.
Yeah. So the lower concentrations seem to be toxic to cells.
Higher concentration is toxic.
The higher
Low
concentration
is not.
Okay. Then, so we think that the higher concentration here is physiology or pathophysiology. Cell viability remains high.
Physiology.
So what you do with your compounds is that in a situation when there is already overload to the cell of nutrients, you create, by alleviating the insulin resistance, the ability for more fats to come into the cell and more fats of glucose to get into the cell. Physiology or pathophysiology?
Sorry, I didn't get your question. Can
you rescue with your compounds the effects of insulin-mediated uptake of fats and glucose.
Yes.
The high palmitate makes you resistant, and you rescue that effect by your compounds. So that means that with the insulin stimulation, you get more of the nutrients into the cell in a condition where the cell was already stuffed with lipids. Right? So the high palmitate condition, you have lots of lipid droplets in your cells.
Yes.
And by using your compounds, you can even get more into the cell.
I think this is just the effects of the compounds to improve the insulin response. Yeah, because in the high palmitate condition, the cells is not respond properly to insulin. They respond completely nothing or less. And the compounds, when you add them to the cell, they can improve this insulin response.
Yeah.
To improve insulin resistance.
Okay, so we proceed that discussion later. Is it okay if I move to the other chapter or not anymore?
I'm sorry.
Okay, good. Yeah.
We have to move forward.
Yes.
So thank you very much for your questions, and we move to the next member of the Corona, Professor Asu. She was also a member of your assessment committee, and she is professor in cardiac physiology of the Maastricht University of Tromsø in Norway, and I would like to thank her that she came all the way today for this discussion, and I'd like to give you the word.
Thank you. First, I want to congratulate you with the presentation. It was really nice, clear and nice, well illustrated. And I also want to thank the university for letting me be one of the members of this assessment committee, and your supervisors inviting me to come here. I also want to start to congratulate you and your supervisors with a really interesting thesis, a comprehensive thesis, and it adds nicely to the important work by this research group on the topic of lipid metabolism in the heart. They are a leading group in this topic, particularly with regard to the CD36 and the role of CD36, and I've followed it for many years, so I really appreciate being able to discuss with you. So I thought that was start by, I mean, you're now working in biomedicine, and there are challenges in biomedicine and basic biomedicine, as we have to choose some models that represents the issue we are addressing. So in your case, it's diabetic cardiomyopathy. So of course, there are several reasons for choosing the model. It could be practical and everything like that. But it's important that we are acknowledge the challenges or the advantages, the strengths of these methods, but also the weaknesses and challenges and limitations of these. And in your thesis, you have used both cell lines, adult cardiomyocytes, primary cell lines, neonatal cells, and adult and animals, right? So I understand from our discussion that you have primarily worked with the cells, and that has been in several of these chapters. I have also experienced primarily from animals, but moved to cells, and it made me think about, okay, some challenges and some benefits. I've realized that there are many benefits and many challenges using cell lines. So I would start by just asking you if you could think about what do you think is some of the benefits of using cell lines, for instance, and also are there any limitations using these cell lines when you address hypotheses related to diabetic cardiomyopathy?
Highly esteemed opponents, thank you for your kind words and your question. Indeed, in my thesis, we used multiple cell lines, but also animal models. But we did the extensive work with primary cardiomyocytes. It has the metabolic feature of a matured metabolism of glucose and fatty acids. So this is very much matched with our hypothesis. And the limitation of using this cell line, like this cell types, is for example, when we test the effects of the TCM compounds, and the next step would be better if we can validate the effects in the cells, in the animal models, to test the whole body effects. But due to the quite strict rules of doing animal study in Netherlands, so it's better to use the cells, because to do in vivo study in Netherlands is quite difficult, and maybe it costs also more than just to use the cell lines. And to use the cell lines, it's also when you have a target, and it's better to do the silencing or knockouts with the cells, and it's very easy to do. But when you want to test the effects for the silencing or knockouts in the animal models, and it costs long time and also much more invests.
Yes, I agree with you on all these issues. There are several benefits related to having cell lines, but also, as you mentioned, they don't mimic necessarily the whole heart, and especially with regard to some of the treatments that can affect the other organs. So you need a model. So you make the model by, I mean, your target is to understand diabetic cardiomyopathy, and that develops by exposing the heart to metabolic stress, we can call it, for a long time, right? So the adult cardiomyocytes and also the HL1 cells, they are exposed then to palmitate, which is a very known model for inducing lipotoxicity. So they are exposed for this 16 to 24 hours. Would you call that a chronic treatment, or would you call that an acute treatment?
For this like incubation time that we did in the cell, in 2 different cell lines, it's overnight, and it's normally with, I think maybe because you cannot really compare, like because when you want to build set up a model for in vivo study, you do like 16 weeks or even longer for high-fat diets feeding. But for the cell type, for the cell lines, overnight or like 24 hours, I think it can be considered as long-term stimulation of hypometase.
But the cells are the same when you isolate the adult cardiomyocytes.
Yeah.
They are like the same cells as within the animal. So we can maybe disagree or agree on that, but it is a challenge.
Yes.
Yes. Secondly, I mean, you use this palmitate, which is a toxic. So it's a model often used for lipotoxicity.
Yes.
So the question then is lipotoxicity the same as diabetic cardiomyopathy?
It is, in some sense, they are the same, but it's not completely the same, because diabetic cardiomyopathy can be induced by different stimulation. Like in the real clinic, not only high fat but also high glucose also cause a problem.
Yeah.
But we use high lipids. In the case of lipid-induced cardiomyopathy, we use high palmitates, and this is a very good model to test the effects of high lipids.
Yeah. So I agree with it. I like the naming it lipid-induced. I can't remember exactly now. More than calling it diabetic cardiomyopathy. I think it's a bit, but I agree it's a part of it.
Yeah, it is
much more stronger. I also want to, I mean, we started doing this ourselves, and for many of the same reasons as you have done. So what we do see in, for instance, we use another cell line. We use adult cardiomyocytes. We have done that. As you have recently showed us with we saw in some of the graphs, the adult cardiomyocytes, they survive nicely these 24 hours, right?
Yes.
We saw that from the survival curve. What about the HL1 cells? Do they, because we know from the literature that we induce apoptosis. It's a common effect of lipotoxicity by inducing with palmitate, is induction of apoptosis. So do they survive the 24 or 16 hours?
We have did the cell viability test in both cell types, in HL1 but also in red cardiomyocytes, and we didn't see like the cell death, like very clear cell death,
Okay.
just on the treatment of high palmitates.
So they survive, these cell lines on your condition, with your conditions, they survive.
Yes.
Okay, I think that could be a very important information to bring in, because if you look at some responses, some glucose uptake or whatever, and the cell actually dies, or insulin response, and the cell is actually dying, so I think knowing that they are actually survive your treatment is crucial.
Yes.
So therefore your lipotoxicity protocol is not very severe compared to some other studies where they do not so yeah. The second thing sorry.
Unfortunately, there is no second time.
Okay,
So
fine.
I have to,
I'm
I
happy
have to interrupt, and I apologize that I have to stop your enthusiasm, but we have to continue. And the next member of the assessment committee is Professor Boogie. He is a professor in immunology and infection of the University of Hasselt, Belgium. Thank you for coming to this morning or this afternoon, and I'd like to give you the word.
Thank you, Pro-Rector. First of all, candidate, I want to congratulate you and the promotion team with this excellent thesis in which you nicely show the importance of the fatty acid translocase CD36 and also the v-ATPase in diabetic cardiomyopathy. But of course, I'm not only here to give you all compliments, I'm here to ask you difficult questions. And my first question might not be that difficult, maybe it is. Can you define a high-fat diet? What is a high-fat diet?
Highly steeped opponents, thank you for your words and questions. For the high-fat diet that we used in the animal study, we took that directly from the company, so it's commercialized products, and we don't have the recipe to make it. But I assume that there will be a lot of fat or different kinds of fat in combination, and to achieve the goal of high-fat diet induced cardiomyopathy.
So what fats are
It's commercialized products, and I am not aware of the recipe of this high-fat diet.
Yeah, okay. But we can agree that most likely there is more in there than just palmitate, right?
Yeah, definitely.
So then I was wondering, in most of the experiments that you do, in vitro cell culture experiments, you only expose these cells to palmitate.
Mm-hmm.
Wouldn't it then be more interesting to expose them to different kind of lipids?
The reason to choose just use high-fat, high palmitate in the in vitro study, because the high palmitate is a well-known stimulant to induce lipotoxicity in the primary cardiomyocytes and also HL1 cells. And the high-fat diet in vivo is also a very well-established method to induce lipotoxicity in the animal model. So there are multiple different kind of lipids we can use, but based on the previous studies, this is a very well established one that we choose. Yeah.
Okay. My second major question is related to what makes a lipid bad. So we're talking about palmitate being bad, but palmitate at the same time is also the most abundant fatty acid in our body, and it plays a very important role, playing a role in palmitylation, for instance. So my question is, what makes a fatty acid bad?
I think, they are very. They also play a very important role in the normal function of the heart cell. So you cannot ignore this. But when you have when the cells is exposed to very increased amounts of fat acids, it's it's beyond the limits of the mitochondrial fatty acid oxidation, so it's causes mitochondrial dysfunction, and this also causes the incomplete fatty acid oxidation and leads to toxic lipid intermediates, and those toxic lipid intermediates such as ceramides or DHA, and this cause the downstream bad effects of this, and this leads to insulin resistance and lipotoxicity. So it's not just palmitate itself.
Is this then the case for all fatty acids? So for instance, omega-3 polyunsaturated fatty acids, they are considered to be beneficial. But if these accumulate, will this also be bad? And they're highly susceptible to oxidation, right?
The oleates that we have also tested before, and because when you give high oleates to the primary cardiomyocytes, and they will accumulate in more the good way, because they leads to increased lipids accumulation, but in lipid droplets, and those lipid droplets is composed by triglycerides, and they are neutral lipids, and it's the storage of the energy, so they are not toxic. That's why the oleate is not as bad as the palmitate. It's because of the toxic lipid intermediates.
Let's see. So in chapter 4, figure 4, which is page 69, you see that your different compounds that they can prevent palmitate-induced contractile dysfunction, right? But in fact, you actually see that contractile function seems to be even higher in those conditions treated with the compound as compared to low palmitate. Might this also pose a problem that these cells are becoming so active? I'm thinking about this will cause a lot of energy, oxygen deprivation, eventually maybe cellular stress, or am I wrong?
It is possible, but the contractility that we measured is we paste the cell with electric stimulation, so they contract at the same time. Just the force is different, so the how to say it? The percentage of the cell they contract compared to the whole length of the cell is different. So they can contract more when you give the compound to the cell. But because we only take the videos for maximum like 10 seconds, so they do that. But I think in the long-term way, it will maybe cause some problems.
Yeah, I'm looking at the proactor. Do I still have time for?
You have time for a question, yes?
Yeah. Okay. So maybe another question is, let's see, so in chapter 4 and 5, you're talking about the different compounds, and these compounds also include flavonoids, right?
Yes.
So how do flavonoids reduce reactive oxygen species? How do they do this?
The reason that we have choose those compounds is based on the beneficial effects in the context of diabetes, mainly type 2 diabetes in the previous research. And they have to be proved to be beneficial to have the antioxidative potential or anti-diabetic potential. But we haven't looked at that in our context. So we know it's beneficial and they have the effects, but we didn't look at.
And do you think that's more like scavenging reactive oxygen species, or do these compounds actually induce an antioxidant response by the cardiomyocytes?
Related to the antioxidative potential, it could be in both sides, because some of the studies have shown that they have the ability to increase the enzymes that involved in both, like because oxidative stress is a balance from both sides, antioxidative or like the other side.
Yeah.
Yeah.
Maybe a final question is,
I just gave you one question, you took three. So there's no final question. I'm sorry to say that. So we're going to continue, and we're going to continue to another member of your assessment committee, Professor Langen. He is involved in the Department of Skeletal Muscle Biology and Lung Pathology at the University of Maastricht, and I'd like to give him the word too.
Thank you, Mr. Prorector. Dear candidate, first of all, my congratulations to the elaborate work that you have described in your thesis and the clear and very focused presentation of today. Of course, my congratulations, I'm happy to extend them to your entire supervisory team. Considering the interest of time, I had a very nice introduction, but I will actually skip this in order to be able to discuss the questions that I lined up with you. And these questions that I have in mind are the ones that I would like to start related to chapter 6, which is a gigantic chapter of 75 pages. I think it's probably the longest chapter I've ever seen in a thesis. And you describe there, in part at least, the role that inflammation has in the cardiomyopathy. And what I would like to do with you is to discuss the mechanisms and the impact of the inflammation that has been described in the heart in those models. And I think what will help us in our discussion is if we have a look at the graphical abstract on page 109, that basically displays the main, well, ideas and to some extent findings that are described in this chapter. And the first part of my discussion with you, I would like to focus on, you see inflammation on the right with, let's say, the orange explosion behind it, and there's two arrows that lead to this inflammation. And one arrow is coming from the diacylglycerol, and the other one is coming from the toll-like receptor 4. What I would like to hear from you is to what extent the diacylglycerol arrow has actually been investigated and shown in this in the work of this chapter, or from what other type of information this link that is shown is coming from. So basically the arrow from diacylglycerol to inflammation, what are the experiments that show that relationship between these two?
Highly esteemed opponents, thank you for your kind words and your question. For DAG induced inflammation, I think there's no direct experiment showing that DH could induce inflammation. The reason that we propose this is mainly because we did lipodomics and we found the DH is the most relevant lipid intermediates in this context. And inflammation is also very obvious in the context of lipid-induced cardiomyopathy. And when you give the NMN treatment or PLAR, and the DH was reduced and also inflammation was reduced. So there's a very strong correlation in between. That's why.
Yeah, exactly. That's the word I was looking for. Correlation or association has been shown. Of course, what we always like to do in cell biology and molecular biology type of experiments is to see if there is a way to show the causal relationship between these two. I know the experiments are not in here, but can you speculate on how you could demonstrate the causal relationship between enhanced DAG levels and the induction of inflammation? Can you come up with an experiment to test that?
I will try. The DH as a lipid intermediates in the context of lipid-induced cardiomyopathy, and maybe to stimulate the synthesis of DAG or the clearing of the DAG, and then test the effects of inflammation could be a way to address this issue.
Okay, so pharmacological modulation some way or another to decrease these DAG levels. Yeah, I agree. I think that would be indeed an approach to follow. Then I would like to move to the other arrow coming from the TLR4 towards inflammation. Can you shortly explain what steps actually make part of this arrow pointing from TLR4 to inflammation that you have shown or that were shown in this chapter? What is the signaling cascade? That's basically what I'm referring to.
For this TLR4, there are evidence in this chapter has been shown that TLR4 is promote the NF-kappa B signaling and also the production of the pro-inflammatory cytokines, and those cytokines and NF-kappa B is very strongly related to inflammation, and this is the reason that this arrow was drawn there.
Yeah, and NF-kappa B is indeed being measured, or a component of NF-kappa B signaling has been measured. I'm not sure to what extent you are familiar with it, but I'm just going to ask. It is the phosphorylation of P65. Is this the best way to measure NF-kappa B activation that you are aware of, or are there also other ways to?
In this chapter, NF-kappa B, the expression is also measured via Western blots and also qPCR, and the phosphorylation of P65 is also measured. So we have multiple ways to measure it.
Yeah, the phosphorylation was used here as a marker of activated. There are also other ways to do this, which I think would be nice to substantiate the involvement of NF-kappa B a little bit more. But then the same question again. So there's activation of NF-kappa B. It's an association with all other effects that you see in this model. Would you know of a way to test whether it's only an association, the NF-kappa B activation, or whether it's actually a causal contribution of NF-kappa B to the phenotype in the hearts and in the cells?
For the exact role of NF-kappa B, we didn't do that in this chapter, but for like TLR4, we indeed use the inhibitor of TLR4 to test the effects of the downstream of inflammation, and it has been proved that this was not only correlation but also is a cause effect of TLR4 induced inflammation. But for kappa B, I assume we can do the same for its role in inflammation.
With what type of tools?
Pharmacological approach.
Yeah, they do exist indeed. That would be
Yes.
an approach. I'm going to kindly look at the prorector whether there is time for a final question.
Yes, there's time for a final question.
Very glad. One of the things that you also propose to be a consequence of this inflammatory signaling is actually the production of inflammatory cytokines. And this is well shown, I think, in the graph of figure 6. My question is, what do these cytokines then subsequently do? So they're produced by the cardiomyocyte, but what is their role in the, because there is already inflammation activated through TLR4, what's the additional effect of these cytokines in the phenotype that then develops in the heart?
The cytokines that produced during inflammation can play multiple roles because it also, as I indicated, also involved in the previous review. The cytokines also interact with oxidative stress, and it may also interact with fibrosis, and this causes the all those interactions cause the final phenotype of
Do they
cardiac.
do this via another inflammatory route, or do they use the same inflammatory route as the TLR4 is activating?
We haven't looked.
You may speculate.
We haven't looked into detail which routes that this TLR4 or this inflammatory cytokines was involved, but it could be just kappa B or those cytokines. It can also be.
I think they may actually further amplify this inflammatory signaling.
Yes.
So I'm satisfied with your answers, and I will gladly return to the corrector.
Thank you very much. Then the opposition will be continued, say be closed by Dr. Broers. He has been involved in the Department of Cell Biology at our university, and I'd like to give him also the opportunity to discuss with you.
Okay, dear candidates, dear Fang. So congratulations. I have to only add to, let's say, the previous congratulations, because I think everything was said already. So the book looks very impressive. It's a very thick one. And also my congratulations to the supervising and also the promotion committee, so all the members. Well, I just got, let's say, were asked to join this committee, so I did not have time to read all the details in your book. And it's not mainly on what issues, but more on technical questions that I would like to ask you. First question is on, which is a method section, actually, in chapter 6. Where you described in methods how you did immune fluorescence with 2 different antibodies. And if I look, so it's on page 134, part 450, and you said you used primary antibodies in mice and in rabbits, and you used secondary antibodies directed rabbit against mice, and what is the other one? The anti-rabbit electrophlora. So, do you think this combination could work? Of course, you have mice antibody, and you have rabbit antibodies, and you have rabbit anti-mice. A secondary antibody. Is that possible?
Esteemed opponents, thank you for your words and your question. I have to admit there are some typos in this part, because to use this secondary antibody is not possible, but we have looked into the methods, and the one that we used is goat and anti-mouse or anti-rabbits. So in this combination, it works. there are some typos.
Okay. Yeah. So it's wrongly written, but experiments put on correctly.
Yes.
Okay, so I'm happy with that. They have another question, and that's on also a kind of technical question. So you used several different loading control antibodies for the immunoblotting. So by the way, you did in one chapter, you did more immunoblottings than I've done in my whole scientific life, I think. So, it's really impressive. And but I have a question on that. So what is your argument to use a specific different loading control for different experiments? So what were your criteria to use it? So for instance, why would you use GAPDH? Because that's used in, well, several immunoblotting experiments. You use GAPDH as a loading control. So what should be the criteria for your loading control? What should it be doing, this particular protein?
For the loading control that we used for immunoblotting, the criteria to select the proper one, it should be like this loading control expression is not influenced by the stimulation that you added or the treatments. So we have tested multiple, and we choose the one that is not influenced by the stimulation, and also based on the methods, because in some cases we measure like the cell presence of this of the protein and also the intracellular parts. So it will be a little bit different in different settings.
Yeah, so I looked it up yesterday, and then I saw a strong advice by, let's say, a company that sells a lot of different loading controls. They say never use GAPDH if you want to study metabolic processes.
Yes, GAPDH is highly related to the glycolysis. So in the metabolic stimulation, the utilization of glucose and fatty acids will be influenced. That's why in my time, and we just use column 3, the other one not GAPDH in this. But we also have to see if the one is influenced or not by the stimulation. So.
And did you also use some other ways to see whether you loaded, let's say, similar amount of protein in your sample?
Yeah, normally we also do the BCE test to make sure that we load the same amount of the protein. But due to the technical variation, sometimes you merit, but for the pipetting area, you can also you may also have some variation there. Thus, another way to solve this issue could be like when you do the Western blots, and you can also quantify the total protein that's it's loaded on the gel, while the chromocy blue staining, so it will quantify the total amount of the protein that you loaded indeed. And then use that as a way to normalize, it will be more accurate.
Okay, so I think you did not do all these blotting by yourself in chapter 6? Okay, yeah. So another thing, so what would you also, so the one would be, of course, let's say, looking at the amount of protein that you load. I think that's a good, let's say, addition to it. What should you also, could you also have done? So, so you use only one loading control. What could you also do? So use only GAPDH, but what would be a good alternative also to prove further that you have the good amount of protein?
To use chromocy blue staining for the total protein.
Yeah, but also.
Horen est.
Dear Ms. Candidate, you are allowed to finish your answer, but you also can decide just to stop. It's up to you.
For a specific protein, we can do the specific control, but also quantify with the total protein with chromocy blue, and for the final normalization. And to combine these two methods, it would be more accurate for the final results.
But also use another
No. She's only allowed to answer, but no more questions anymore. I'm sorry. So, Ms. Candidate, Ms. Wang, the time appointed for defending your thesis has now officially passed. The degree committee will now stay here. You will withdraw. And we're going to discuss the quality of your thesis and the way you have defended this thesis this afternoon. And I request that you and your company leave this room and wait outside till the beadle will tell you that you come back to the office over here. Thank you very much.
Please all rise.
Hours? Usually they are protocol of 1 hour maybe.
U grote pak.
Something that's specially
Very special.
Iedereen.
All rise.
Iedereen.
Ja, maar ik ben pas gegaan, dus dat.
Please be seated.
Dear Ms. Wang, the degree committee here present has assessed not so much the quality of your thesis, that was approved, but we looked the way you have defended your thesis, and we also looked at the quality of your presentations. In view of its positive verdict, but also taken into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Congratulations. Professor Klatz is authorized to confer upon you this academic distinction in accordance to Dutch University customs, and therefore I invite your promoter now to take the floor.
Please advise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Fang Wang, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university.
Is there a photographer available? Then you should come forward, please. Okay? Yes, good. Okay. You have to make the picture.
Yes. Okay. Please be seated.
I'd like to give the word to Dr. Luiken, who's going to give the laudatio to the new young doctor. You may stand up.
Okay. Dear Dr. Wang, dear Fang, I'm happy to congratulate you with obtaining the predicate doctor. It has already been more than 4 years ago that your adventure in Maastricht started. Time goes quickly. You started with studying the potential of selected compounds from traditional Chinese medicine to reverse lipid-induced insulin resistance and contractile dysfunction in the heart. And as to whether these compounds operated via restoring endosomal acidification and internalizing the fatty acid transporter CD36 back to the endosomes. Indeed, we found that all selected TCM compounds induced CD36 internalization and restored contractile dysfunction. Further, some of them reversed inflammation and others corrected oxidative stress. But these effects were inconsistent. While most of them did not restore endosomal acidification, 1 or 2 indeed had significant beneficial effects on the endosomal pH. I repeat, really interesting. Unfortunately, there was no time to follow this up because the study ended up with taking the full 4 years, and this is something for further research. Despite that you were good in lab work, including still culturing, never an infection, and complicated pipetting schemes, you did not like it too much, and that is why you initiated a network pharmacology approach using several literature databases to share the different view on via which mechanism our selected TCO compounds exert their beneficial effects on the lipid-overloaded heart. You have initiated the study without help of the supervising team, given that we all lacked more or less the background knowledge of working with large literature data sets. In the end, this literature study yielded some potential interesting target proteins. The next step should be to verify these targets in our heart cell cultures under high lipid exposure. Then, something unexpected happened after 2.5 years. You became pregnant, and in February 2023, Xila was born. I met him a few times, and he seems like a happy little guy. For you, it must have been difficult to combine a baby with the work in the lab, but nonetheless, we carried on, and after your return from pregnancy leave, you could participate in a big study from my former PhD student Fang Wang, who has now returned to China already for 4 years and has stayed a friend and close collaborator. He also tested several compounds on reversing lipid-induced cardiomyopathy via restoring endosomal acidification, including the by us developed KLR cocktail. This fitted very well in your research line, and you contributed to the study with invaluable radioactive tracer studies and other in vitro experiments. This study has now been published in the Circulation Research and fills up a lot of space in your thesis. Finally, and here I speak in name of the whole supervising team, I wish you the best in your personal life and in your career, and also with your husband and the little smiley guy. Where is he? Outside. Congratulations. I give the word back to the prorector.
Yes, and of course we're going to applaud. And I think you are allowed also to bring the young child.
Yeah.
Well, he's already 4 years old. He should enjoy too. That's with us.
Yes.
Yeah, okay. You see? Very nice.
Little smiley guy.
You can join your wife if you like. Yes. So basically, I would like to congratulate the new young doctor. How does it feel? Feels good?
Yeah, that's good.
Feels good. I would like to also, on behalf of Maastricht University, our board of directors, and you did a very nice job. Basically, your degree now is in our country, in the Netherlands. Your major and your last and final academic distinction which you have and you receive. So it's a high opportunity. And it's also important because you are, I would like to address that, you are a CSC student, and basically we're always proud of all the CSC students. Some go to China, some remain, but you all are our ambassadors worldwide, wherever you go. So keep that in mind. My congratulations also going to the promotion team who guided you in these 4 years. And it hasn't been mentioned here, I was a little bit surprised about that. You had a difficult time because you did your thesis in Corona times. So there was basically a time when there was no labs, no experiments, and of course you're going to do a lot of databases investigation. That is related to this work what you did. So, and you did a nice job about this. I also would like to thank the members of the Corona who are here, and you had a large number of different kinds of questions, and I always mention that they just have 9 minutes, but it takes more than 9 minutes to do the preparation for all these questions. And I would like to thank all of them to be here today at this event. And for the other students here, the CSC students, we're looking forward to see you also here appearing in the near future when you're going to defend your own thesis. And I think that is basically, you saw a very nice example today. Congratulations. And by that, I'm going to close officially the ceremony, and therefore I have my hammer over here. And basically, this is first. How do we proceed? We as a team